two scientists who created the structure for DNA, Mr. James Watson and Mr. Francis Crick. How are you doing today, James?
I am doing well, thanks. How are you today?
I am also well, thank you. And how are you, Francis?
I too am well, living life to the fullest.
That's great to hear. So, how did you guys first meet?
I met my colleague Francis in the year of 1951 at the University of Cambridge, where we composed many different experiments in the Cavendish lab.
Please describe your most famous experiment, the structure of DNA and the double helix.
After returning from Rosalind Franklin's lecture in 1951 on DNA existing in two forms, Francis and I composed an experiment, but the model we had created had failed. We failed many times, but we would never give up. Then we took a crucial step, suggesting the molecule was made of two chains of nucleotides, with one going up and one down. After also learning about base pairs from Chargaff's findings, we added to the model so that the matching base pairs interlacked in the middle of the double helix. To keep the distance between chains constant, we showed each strand of the DNA molecule was a template for the other. During cell division, the two strands separate, and on each strand a new other half is built. DNA can then reproduce itself without changing its structures.
Briefly describe the model that was actually created in the lab.
We used four brass templates illustrating the base pairings of adenine and thymine, or cytosine and guanine. Two templates show the correct base pair shapes. Two others are earlier misconceived models. The measurements were 2 centimeters by 24 centimeters by 8 centimeters.
How and why was this experiment so important?
The experiment itself was revolutionary and has been called the most important biological work in the last 100 years because of how important DNA is today. DNA may be the most important molecule in the world today. Finding the structure of it is extremely important as well. It was the only way to determine whether they did in fact carry genes.
Describe the actual structure of DNA itself.
DNA is made of nucleotide, with each nucleotide containing a phosphate group, a sugar group, and a nitrogen base. The four nitrogen bases are adenine, thymine, guanine, and cytosine. The order of these bases is what determines DNA's instructions or genetic code. Nucleotides are attached together to form two long strands that is called a double helix. The two bases on one strand pair with bases on another strand. Adenine pairs with thymine and guanine pairs with cytosine. DNA molecules are so long in fact that they can't fit into cells without the right packaging.
Alright, it was an honor talking to both of you, and thank you so much for your time.
We both appreciate you having us here. Thank you for everything.
Chris Wagner signing out here on the structure of DNA, and have a great day.